In life science companies, scientists spend a day a week on repetitive and manual steps for analysis and reporting. And because all this manual work needs to be checked and verified, managers lose even more time and precious resources. DataChaperone provides a platform that we fill with your workflows so your analyses become standardized, automated, traceable, so you can focus on your innovations. Nova will show you an example.
Now I'm inside the DataChaperone platform. Today I'm performing sterility testing for batch release and select the workflow for plate statistics. First, I provide a run name and upload my filled-in experiment design template. From this file, DataChaperone understands the setup of my experiment and guides me step by step, asking for the right data files to be uploaded. A short explanation reminds me what this analysis is about, so it never feels like a black box. I define my condition label, in this case concentration, confirm the control, and choose which samples to include in the analysis. Before running, I review the configuration. If I spot something that needs adjusting, I can easily go back, make the change, and continue. Now the analysis is started. In most labs, this would mean hours of manual work, sometimes spread over several days, with multiple people double-checking every step. On DataChaperone, the results are ready immediately. In the results section, I see interactive graphs. I can hide or show lines or focus on a single sample. I can even adjust the plot, like changing the axis scale, plot title, and axis label, and save these edits instantly. Beyond these visualizations, DataChaperone can also generate standardized reports and ELN entries automatically. You'll find examples of that in our other videos. Let me show you how easy it is to browse results from different analyses across workflows. This makes it simple for teams to reuse past results and learn across projects. With free text search, I can return to today's sterility test results, instantly bringing up the analysis we just completed. The workflow panel also lets me navigate through every step of the process: the experiment setup, configuration, raw and merged data, process data, and finally the results. Every dataset can be exported as CSV, results can be downloaded. So now you've seen firsthand how tasks that would normally require hours of repetitive spreadsheet work are completed automatically, reproducibly, and immediately, freeing scientists from manual effort and giving managers results they can trust.
What you've just seen is how we automate an entire data analysis workflow. For managers, this means that scientists reclaim a day a week to focus on science instead of spreadsheets. Turnaround times shrink from days to minutes, and you can finally compare between batches, see QC trends, learn from your data. If this resonates, I'd be glad to talk to you and see what we can do for your lab.